La siguiente ponente de la sesión de hoy se llama Pilar González García y nos va a presentar su trabajo titulado Coenzyme Q10 Regulates Sulfide Metabolism and Pathway Connected to One Carbon Metabolism Involved in Mitochondrial Dysfunction. Cuando quieras, puedes empezar.
Vale. Hi, everyone. I'm going to do the presentation in English. My name is Pilar González. I work in the Biomedical Research Center of the University of Granada, and we look for possible therapies in mitochondrial dysfunction. Today, I'm going to show you our last results that shows how coenzyme Q regulates sulfide metabolism and other pathways involved in what is called as one-carbon metabolism. It's important to know that these pathways recently have been associated to mitochondrial dysfunction. The mitochondria, as you know, plays a lot of different roles in the cell. The most important of them is the energy production. This bioenergetic process takes place in the inner mitochondrial membrane, in what is called as oxidative phosphorylation system, or OXPHOS system. In this process, complex 1 and complex 2 transfer electrons to the coenzyme Q. And at the same time, a proton motive force is produced in the intermembrane space. This is the force used by the ATP synthase to produce ATP. Coenzyme Q can also transfer electrons from other different mitochondrial enzymes. I'm going to highlight the SQR, an enzyme involved in the hydrogen sulfide metabolism. Due to the important role of the mitochondria in the cell, a problem in this point is the origin of what is called as mitochondrial dysfunction. Mitochondrial dysfunction can be produced for a mutation in any of the genes that encodes for mitochondrial proteins. And I'm going to focus attention on mutation that encodes essential proteins in this OXPHOS system, especially in complex 1 and in coenzyme Q. The mitochondrial diseases are clinically heterogeneous due to the participation of many different pathways and mechanisms. In the last years, the folate cycle and nucleotide metabolism as the transformation pathway, this last one is involved in hydrogen sulfide metabolism, have been described as pathomechanism involved in mitochondrial dysfunction. At the same time, there is studies that shows a modification in hydrogen sulfide metabolism in pharmacological inhibition of complex 1, and it has been described in CoQ deficiency and other different mitochondrial disorder, where there is a decrease in the coenzyme Q levels, that there is a decrease in the SQR levels, the enzyme linked to the coenzyme Q. And this produces a disruption in the oxidation pathway with an accumulation of hydrogen sulfide and a depletion in glutathione system. However, we don't know how these pathways are regulated and how they interplay one with each other. For most of the cases, the therapeutic option is limited to palliative care, but CoQ supplementation is recommended for most of the patients. With this data, we have two hypotheses. The first one say that if we increase CoQ level with supraphysiological levels of CoQ, there could be a modification in the SQR levels, and this could modify other enzymes of the sulfide metabolism, the ones from the transsulfuration pathway, and these changes could produce changes in other pathways from nucleotide metabolism, folate cycle, or serine synthesis. These three last one participate in what is called one-carbon metabolism. And in a second part, we think that these changes could be modulated by the sulfur amino acid availability, because this is a source for hydrogen sulfide. So we decided to perform a study to check in different models with mutation in complex 1 and CoQ how CoQ supplementation change this pathway and how this pathway change with sulfur amino acid availability. So we designed an in vitro model with 7 different cell lines with mutation in complex 1 and 2 cell lines with mutation in CoQ. We treated the cells with 100 micromolar of CoQ10 for 7 days. And after the treatment, there is an important increase in the CoQ levels in all cell lines. First, we decided to do a transcriptomic profile in 2 cell lines in control and in 1 mutant in complex 1, the one with a mutation in the NDUFS1 gene. This transcriptomic profile shows that there are a lot of different canonical pathways that change with the treatment. In the control, in graph A, and in the mutant in graph B. It is important to say that between the 20 and the 35% of these changes are related to sulfide metabolism and 1-carbon metabolism. Further analysis of this transcriptomic profile shows that in sulfide metabolism, there is an overexpression of the SQR, the enzyme from the oxidation pathway, and there is a downregulation of the enzyme CSE and CBS, 2 enzymes from the transphosphorylation pathway. In the serine biosynthesis pathway, there is a general downregulation, and there is an imbalance of the genes that participate in folate cycle and in nucleotide metabolism. I'm going to highlight that these changes with the treatment with CoQ are the opposite as the ones described in mitochondrial dysfunction. So it looks like the treatment is restoring the function that was altered in the mitochondrial disease. In this graph, you can see the relation between all these canonical pathways and the enzymes and protein involved. We continue with gene expression analysis, and we focus attention in the hydrogen sulfide enzymes, the ones from the oxidative pathway, the SQR, and also the SUOX and the TST, and the enzymes from the biosynthetic pathway of the hydrogen sulfide metabolism, the CBS, the CSE, and the 3-MST. We perform gene analysis in all cell lines I described before, with mutation in complex 1 and in CoQ. In the SQR mRNA levels, we have the same results as the one in the transcriptomic file. There is an increase of the expression with the treatment, as it is a disruption in the expression in the enzymes from the transsulfuration pathway, the CBS and the CSE. We can't see any change with important change in the expression with the treatment in the other enzymes, in TST, in SUOX from the oxidation pathway, and in 3-MST in the transsulfuration pathway. So here we can say there is an inverse relation between SQR mRNA levels and the mRNA levels from the enzyme of the transsulfuration pathway. To see if these genetic changes produce a change in the protein level, we decided to measure the protein levels of the enzyme of the oxidation pathway. Here we have the results of the measure of the protein levels. If you see graph B, you can see that the protein levels with the treatment increase after the CoQ supplementation in all groups. So we have a correlation also here with the transcriptomic files, while there is no changes in the other enzymes, in TST and in SOUX. These results were obtained in vitro, so we also try to check it in vivo. For that, we use 3 different in vivo models. In the first one, in graph E, we use wild-type mice treated with CoQ, where there is an increase in CoQ levels and in SQR protein levels after the treatment. In graph F, we use an in vivo model with complex 1 deficiency, because for a mutation in the NDUFS4 gene, in this case, the CoQ levels are similar if we compare to the control, but there is an increase in the SQR protein levels. In the last graph, in graph G, we work with in vivo model with CoQ deficiency. In this case, the CoQ levels decrease in the model as the SQR protein levels. And these changes were the same if we treated the model with NAC, with an acetylcysteine, or with a diet with sulfur amino acid restriction. So the changes we observe in the oxidation pathway don't depend on the sulfur amino acid availability. in the enzymes of the transsulfuration pathway, we also have a correlation with the transcriptomic profile. The protein levels of CBS and CSE also decrease after the treatment with CoQ. And in the in vivo model, there is a decrease in the models that have higher levels of SQR, like the wild type treated with CoQ and the mutant in complex one. And there is a tendency to increase the protein expression of CBS in the model of CoQ deficiency, as it was the model with lower levels of SQR. To check if these changes in the transsulfuration pathway were directly produced by CoQ or produced by the overexpression of SQR, we decided to overexpress SQR in 3 different cell lines, in control, in 2 mutant in complex 1. And we check mRNA levels and protein levels for SQR. In graph A, you can see how the cell treated shows an increase in the expression of SQR, how it was expected, as an increase in the protein levels. At the same time, in graph B and C, you can see how this treatment, the expression of SQR, produced a decrease in the expression of CBS and CSC in all cell lines, and the same happened with the protein levels. We also check if the effect of CoQ supplementation in 1-carbon metabolism was produced also by the overexpression of SQR. So we measure some of the genes involved in these pathways, and we have a very similar correlation with the transcriptomic profiles. So as a conclusions, we can say that supraphysiological levels of CoQ, it means a CoQ supplementation, produce an increase of SQR, the enzyme from the oxidation pathway. And this change the expression of the enzymes of the transsulfuration pathway that are connected to the pathways from the 1-carbon metabolism. So the increase in the levels of CoQ10 seems to restore the function that was modified in the mitochondrial dysfunction. For that, CoQ supplementation could be a good therapeutic option for mitochondrial dysfunction, or in other patients with problems in hydrogen sulfide metabolism, or in nucleotide metabolism, folate cycle, or transsulfuration pathway. At the same time, because CoQ seems to improve the sulfide oxidation, this treatment could be an option for patients with epimalonic encephalopathy or Leigh syndrome due to mutation in SQR, because in this patient there is an accumulation of hydrogen sulfide and a depression in the glutathione system. But also it could be a good idea for patients with Crohn's disease, colon cancer, or thyroid carcinomas, because in these cases there is an upregulation of the transsulfuration pathway. So I'm going to finish with my presentation, but I would like to say all of you, thank you for your attention. Thank you also to the organization of the Congress to select this work for our presentation. And especially thank you to the people from the group, from the lab, for their work and ideas. Thank you so much.
So thank you very much for your presentation. It has been very interesting. And I wanted to ask you something.
Okay.
Do you prefer that we speak in English or in Spanish? Doesn't matter.
Mind. I don't mind.
Okay, so I wanted to ask you something. Have you thought to study the metabolism with the metabolomics, for example? You have, yes, you have speak about genes, and I was waiting something more, and you have speak with related with the proteins. But I don't know. Have you thought to study the metabolism?
Okay, this work is the first step. So we start with the transcriptomic. We already did some experiment about metabolites. In fact, we checked the glycine-serine ratio to see if the transcriptomic changes were really a really metabolomic change. And we checked that there was an important change in glycine and in serine synthesis, but we didn't continue with the metabolomics. We focused attention on the proteins. But it's something that we have in mind that we have to check. Yeah.
Okay. So thank you very much.
Thank you.
Vamos a dar paso a la siguiente ponencia.